So when it is about the introduction to bioinformatics, we need to understand the term bioinformatics first. And when we break the term, you will see the meaning of the term itself. So in the beginning, when this term was coined, there was like the biologists used to generate the large amount of data. But what was the issue? The issue was where to store that data and how to analyze that data. What happened? So at the time when the term bioinformatics was coined, it was the biology plus information technology plus mathematics and statistics. That means the biological problems used to be solved using the information technologies by applying the mathematical and statistical approaches. Okay. So, but nowadays it is not necessary only for the biology, whatever the, as we are in the era of multidisciplinary. So whatever your discipline, you just understand your query and you have the, if by learning the bioinformatics skills, you will be able to solve those problems. So when it is about the bioinformatics, in the beginning, we can do the biology, chemistry, computer science, statistics, mathematics. These are the disciplines, few disciplines that combined and the term bioinformatics was coined. But as we can see, as we have discussed, that from any discipline, you can access the bioinformatics. How there are few applications we can discuss here. Just wait. Few applications of bioinformatics. Definitely, this is not the complete list. We will be discussing few of them. So very first thing, the sequence analysis. As we know nowadays, this is the widely used approach that has been used. The sequence can be generated by biologists, but definitely for the analysis, you need the bioinformatics skills. The drug designing. When we talk about the drug designing, if we go back in the previous time, we will see in this drug designing, it took like 15 to 20 years for a single drug to launch into the market. But in the bioinformatics time, what the screening of the drugs, the compounds become lesser time, we can do the screening in the less time as compared to the previous time. This is how the bioinformatics helped in the drug designing as well. Likewise, the gene expression, proteomics, genome analysis, immunoinformatics, everywhere the bioinformatics approaches can be applied. And these approaches, you can generate your own approach as well as you can use the already defined approach. Next, as we discussed, it is not limited to the biology only. There are many subfields and the related disciplines of this bioinformatics. So you can include the proteomics, the metabolomics, system biology, pathway analysis, molecular modeling, which we call the structural biology, basically, you can say, the transcriptomics, phylogenetics. So all these are the disciplines where the bioinformatics approaches can be incorporated. And by understanding this, we understand the importance of the bioinformatics skills. So if you are having any bioinformatics skill, if you understand how to access the, whatever particular field, we can say the structural biology, system biology, whatever, you want to solve any of the question related to any of these discipline, your bioinformatics skill would be helpful to solve these questions. Next, we will be discussing the scope of bioinformatics. Definitely, this is not the complete list, but when we broadly discuss the scope of the bioinformatics, what comes in our mind, as we have discussed in the earlier time, the biologists used to generate the data, but biggest problem was to store the data and to analyze the data. So bioinformatics came into the market with the help, with the solution to provide the facility to store the data, to fetch the important information from data. Okay. In the continuation to this fetching and processing of the data, the very first thing, that is the management and the analysis of the large set of biological data. One example is the NGS. Okay, we will be discussing in the next slide. So this management and analysis of large biological data, this biological data could be anything, definitely. The computational coding for the several applications. Here, the computational coding refers to the programming, basically the programming skills also incorporated into the bioinformatics. So, and with the help of bioinformatics skills, you can generate your own, you can develop your own diagnostic tools as well. Okay, as we have discussed, you can use already available tool as well as you can design or develop your own tool. And drug discovery, that is the biggest field. You can say this is the field where the bioinformatics is like a miracle. And the personalized medicine, one part of the drug discovery itself. So in the continuation to this, we will be discussing few approaches where the bioinformatics is a key point. So first of all, we'll be discussing the next generation sequencing, as this is the widely used subject nowadays. So many of you might be knowing what the NGS is. We will start with the brief definition that it is the high throughput method which is used to determine the portion of the nucleotide sequence of an individual genome. Okay. Here, there are few advantages of NGS. Why NGS? Why not? There are many other sequencing techniques are also there, like the Sanger is one of them. Why NGS? Because it is fast. It is capable to detect low frequency variants as well. It is definitely cost effective, and it has capability to cover the comprehensive genomics. Okay. And next, when we discuss about the application of this NGS, it helps us to analyze the gene regulation. It helps us to discover the mutation in the genome. It helps us to understand find the molecular diagnostic for the inherited disease. We can discover the biomarker as well, and many more. We can identify the drug target with the help of sequencing using the NGS approach, definitely. But when we talk about the sequencing, it is not limited to only one thing. You can do the sequencing of DNA, you can do the sequencing of RNA, the bisulfite sequencing, epigenomics, all these things are there. And when we are going towards, for example, when we're going towards the DNA sequencing, what kind of analysis can be done? We can find out the point mutation, we can find out the small insertion deletion, we can go for the copy number variation and the structure variation as well. Okay. So this is when we are going towards the DNA sequencing. In the same way, and the RNA sequencing help us to find a differential expression between the normal and the diseased. It helps us to find the gene fusion, alternative splicing, RNA editing. So these are the things, these are the points that can be analyzed using the sequencing. But again, how would you analyze these things? How this data can be analyzed? The sequencing technique will give you the sequence. That's it. That sequence need to be analyzed where there you need the bioinformatics skills. Nowadays, if you will see, most of the NGS tools or softwares are developed by a bioinformatician. Like on the daily basis, they are upgrading as well. So before sequencing will help us to do this kind of analysis, but this analysis and interpretation can be done with the help of bioinformatics skills only. Okay. Next approach, we are going to discuss the single cell sequencing. As we know, as we have discussed the NGS in the previous slide, well, for example, the RNA sequencing we are discussing. So what happens when we do the sequencing? We are, for example, we are concerned for particular tissue. For that particular tissue, we are collecting the sample, and whatever the sequencing, whatever the output is coming out, that output is somehow the average of all the cells. Okay, but in the single cell sequencing, the concern was that each cell has its own role in the disease or in the whatever the thing we are studying. So this single cell sequencing, it provides the cell specific genetic information. Okay, so we can say it is the process to examine the sequence information from individual cells. As in the other NGS techniques, we use the bulk cell. It is the bulk technique. It was a bulk technique. And here in this figure, if you can see, it is like briefly explained here, whatever your query tissue for a kidney, liver, whatever, if you are curious to understand the tumor at this tissue, what will happen? We have to isolate each and every cell from this tissue, and these cells will be like some barcode will be allotted to these cells, and then the sequencing will be done. And that sequencing, after the sequencing, the processing and the pre-processing and processing of the data will be done. Again, here the bioinformatics is required. You can do till barcoding and till sequencing using the biologists and the other approaches, but analysis again require the bioinformatics skills. There are many packages, many tools are available to do the single cell sequence analysis. So the main purpose of this single cell sequencing was to study the normal physiology and the disease in the mammalian system. And the application, in the first application, we are going to discuss what kind of this single cell sequencing can be done. The DNA methylome sequencing, it helps us to quantify the DNA methylation. The transposase accessible chromatin sequencing, that is the method which helps us to map the chromatin accessibility across the genome. And the transcriptome sequencing, it is most widely used in the single cell sequencing, and it helps us to analyze the RNA expression. After this, along with these applications, like there are what are the medical applications of the single cell sequencing? We understood the concept why the single cell is in the market, but why it is required to understand these medical applications. It helps us to understand the cancer, reproductive system, immune system, nervous system, the microorganism. Like single cell sequencing can help us to for the detailed study of the disease. For example, we can take an example of cancer. So what we are seeing here, whatever the genetic variations in a cell with different genetic and phenotypic characteristics, whatever the tissue we are considering, the high degree of the heterogeneity may be related to the mechanism of the tumorigenesis that can be concluded from the analysis of the single cell sequencing. The major approach is that is the metagenomics, that is again the sequencing only. What is in the metagenomics? It is the genetic analysis of the genome which contained with an environmental sample. Okay, so it is the study of the structure and function of the nucleotide sequence which are isolated, and whatever the bulk sample is there, this bulk sample could be soil, water, and human skin as well. Okay, so this metagenomic sequencing is better in detecting the novel, rare, and unexpected pathogens. Okay, and what are the applications to this metagenomic approach? It helps us in the identification of the unique bioactive compounds, antibiotic, and enzymes as well. With the help of metagenomics, we can also identify an unknown pathogen in the outbreaks of disease, definitely. And it also helps us to determine the gut microbial species, and it also allows to monitor the human health. So these are the benefits, these are the application of the metagenomics. Again, the bioinformatics skills are required to understand the data, to fetch out this information and analyze the data. Next, we will be doing the structural bioinformatics. In the beginning, we have discussed the importance of bioinformatics in the drug discovery, and structural bioinformatics is part of that. Structural bioinformatics, the name is indicating here, we will be analyzing the 3D structure, basically the processing of the 3D structure of the biological macromolecules. So when it is about the structure can be solved by any approaches, any experimental approaches. But if the structure is not available, bioinformatics also help you to model the structure. Okay, you can model the structure, whatever the structure that is not. So that structure could be for protein, for ligand, for any small compound, anything. Okay, here the PDB, MMDB, PubChem, Zinc, these are the databases or the repositories, you can say, where the structures are stored. As in the beginning, we have discussed what was the major aim of the bioinformatics: to store the information. Okay, whatever the structure that has been generated, that need to be stored so that further that can be utilized. That is the importance of bioinformatics here. So along with these repositories, there are many more other repositories are also available to store the 3D structure. Now, this structural bioinformatics help us to do the drug discovery. That is like it plays a vital role in the drug discovery. How? It helps us in the target validation. If you will have the knowledge of the structure, if you will be able to visualize, to understand the structure, you can validate your target for the disease. And active site identification. This active site identification is something basically, I'm not going in the deep, but you can say whatever the structure you are considering for whatever the protein, and if you have modeled that structure, you don't know where the ligand can bind, where the compound can bind, and this active site identification approach can help you to find where exactly the ligand or the compound can bind to inhibit the activity of the protein. Lead optimization, drug likeliness properties, prediction, all these are the part of the structural bioinformatics. So basically, the aim to do the structural bioinformatics is basically to have that deeper knowledge about the structures. And if you have to work in the drug discovery, the very first thing that is understanding of the structures of the macromolecules. Okay. Nowadays, this artificial intelligence and machine learning are the hot topic, definitely. In the bioinformatics also, these approaches can be applied. So these are the approaches basically which are used to examine procedure and classify the biological data in the short time. That is what the purpose of these approaches here. Machine learning is nothing but the subfield of the artificial intelligence, where it aims to mimic the intelligent abilities of human by machines. Here in the combination of this machine learning in the bioinformatics, we can do the evolution that is the phylogenetic studies. Okay, with the help of these machine learning approaches, we can construct the phylogenetic tree. Definitely, the text mining, it helps us to extract the knowledge from the biological database, as well as it allows to processing of the large text bodies as well. Next, on the proteomics, in the proteomics where we study the proteins, in the this machine learning approaches in bioinformatics help us for the protein interaction prediction. It also helps us to annotate the protein. It can also help to find out or predict the structure and function of the protein. In the genomics, it helps us to find out the gene function prediction. It also helps us to find out the motif identification. What is that? The concerned part, basically. Microarray data analysis, where we analyze the differential expression of the disease and the normal, and the coding reason identification, splice site prediction. There are many more, definitely. And in the metabolomics also, it helps us for the targeted metabolomic profiling. It also helps us for metabolomic simulation programs. So whatever we have discussed here in this machine learning thing, so what my aim was, there are already tools are available to do so. Okay, whatever tools are available, you can use them as well. You can, as well as you can create your own. That is the power of bioinformatics. So you can customize your own tool by learning the bioinformatics skills that will help you to whatever your problem is, to customize the solution regarding your problem. You to create your own tool, your own algorithm, own program using. If you understand the skills, you can do anything. So here I would like to conclude bioinformatics as one of the major field which is growing at the faster rate, definitely. And there are many experts who have sought their career in the bioinformatics, and besides the gaining lucrative income, they are also gaining the experience and knowledge as well. Okay. Bioinformatics definitely it is expecting many fold faster than any other field of the biology. Okay, and this growth rate is just because the research by expert in this field. So whoever the expert of the bioinformatics, they are very active. They are doing research on a daily basis. And as we have discussed, on a daily basis, the tools are upgrading. Today, we are using one tool. In the next 10 days, we are getting another tool for the same analysis. That is the more updated version. Okay. Bioinformatics also made the research methodology much simple and easy. Okay. Other fields also desire to see the advancement of bioinformatics. Why? Because if the bioinformatics will be advanced, their queries, their problem will be solved. Okay. So this is what, like, bioinformatics skills will help you to understand and to solve your problems. If you have the bioinformatics skill from whatever disciplines you are, you can get the solution, definitely. That is the power of bioinformatics in this time. Now, thank you so much. I hope the concept bioinformatics might be clear in your mind after this. If you have any question, you can ask me.